So chapter 13, so we discussed this, we will discuss the molecular diagnosis of chromosomal disorder. So we will introduce you all the technique that have been used to detect the chromosomal defect disorders, all right? So the chromosome, okay, chromosome, for the chromosomal disorder, you obviously you need to look at the chromosome. And in the metaphase that we can see, as you remember, the metaphase, the mitotic chromosome can be distinguished by their relative length and the location of the centromere, right? The centromere, that's the region of a chromosome responsible for the segregation at the mitosis and the meiosis. And as you know that during the mitosis, the centromere moved to the pole, right? To the pole, right? Opposite pole. And the attached chromosome appear to be dragged along behind it. Okay. So that's the centromere, and basically the place where the chromosome attached to this microtubule, we call it we call this a spin, the kinetochore, right? It's kinetochore. Okay, that's how they drag this chromosome to the opposite pole, and finally then separate the two cells, right? So metaphase, you will see all the chromosome listed in the they condense in the cell, right? And then because of the mitotic chromosome give us this put all the chromosome together, and then you can see the shape. So the technique has been developed to examine the chromosome, and this technique is called karyotype. So the karyotype can help us to display the chromosome by length, and then it can display the chromosome by the position of the centromere, all right? So the karyotype. So basically, the karyotype can be defined as the accurate organization of the chromosomal content of any given cell type. And in the karyotype, as I mentioned, that can display chromosome by length. So the karyotype, those chromosomes can be arranged and numbered by size from the largest chromosome to the smallest chromosome. Okay, and karyotype is the normal nomenclature used to describe the normal or abnormal constitutional or acquired chromosomal complement of an individual tissue or cell line. Okay, and as you can see here, this is the human karyotype, right? And this one is the normal chromosome. You don't see any defect, chromosome defect. And then they arrange the chromosome from the largest, which is chromosome number 1, to the smallest, the chromosome number 2. And then we have sex chromosome X and Y. Okay. Yeah. Any question? No.
No,
And so to get this karyotype, basically you have to grow the cell. And then you grow the cell, then you arrest the cell at the metaphase stage. To arrest the cell at the metaphase stage, you have to treat the cell with this chemical cosmine. So once you treat the cell with cosmine, all the cells are fixed at the metaphase. All right, so that gives you all these chromosome morphology. And then you have to stain the chromosome so that you'll be able to see. So basically you treat fix the cell with trypsin, and then you add a dye, the giemsa, all right, the giemsa. And so you will see these chromosomes. And because it's a giemsa, so we call this chromosome specific band. Now you see this band here, we call these G bands, all right, G bands. And also this karyotype, not only to display the chromosome by length, also can display chromosome by position of a centromere. All right. So for example, centromere. Do you see the position of centromere? And the centromere, basically, remember that we learned this centromere basically are the region that you have the highly repetitive alpha satellite sequence. Okay, that's the centromere. And then we have this metacentric, all right, metacentric. The chromosome arm are relatively equal in length, all right, for this P1 and Q1. And then submetacentric. So in the submetacentric, you can see one arm is obviously longer, much longer than the other arm, all right? That's submetacentric. And also we have the acrocentric. Acrocentric, okay, one arm is extremely short, all right? It's extremely small and short, and the other arm is extremely long. That's called acrocentric. Also, we have the telocentric. The telocentric, basically you lost the one arm. You lose the one arm, okay? One arm is missing completely. So from the metacentric, submetacentric, acrocentric, telocentric, that you will be able to see through the karyotype, okay? And normally they name this, they put the shorter arm on the top, and then we give it the P arm, and then longer arm in the bottom, and then we named it as a Q arm, okay? It's Q arm. And once you learn those P arm, Q arm, then this is a further nomenclature to tell you how they name the chromosome region. So for example, they put the shorter arm on the top, longer arm in the bottom. So it's a P arm here and Q arm here. And then you have different region. And then they say if you want to refer to this orange region, then you can read it. So it's from the P113, so that people will know that will refer to this region. And if a P221, that'll be this region, right? And the Q, and then people know that's in the bottom, the longer arm, okay? So the G bending allow each chromosome to be identified by its characteristic bending pattern. So these are the bending pattern, all right? So the bending pattern can distinguish chromosomal abnormalities or structural rearrangement, like a translocation, deletion, insertion, and the inversion. And so that from here, we know this G bending that can help us to divide those pattern into the region, bands, and subband, okay? So that'll be a region, band, subband, okay? And so that's the one way that you can see those metaphase chromosome bending, right? Can have a G band. Also, you can have a R band, right? The R band that basically you have a very harsh treatment of chromosome at like 87 degrees for 10 minutes before you apply the Giemsa, all right? Apply the Giemsa. Then you can have this R band. The R band basically can stand the chromatin region, okay? And then C bending. C bending basically that will stand the centromere. The C bending centromere. Like here is the C bending. You can see the C bending. So that give you this stand of the centromere. See the difference between C bending and G bending here. G bending, you can see all this bending pattern. For C bending, you don't see the pattern. You only see this centromere. I tell you the location of the centromere, okay? So basically, C bending or centromere bending results from the alkali treatment of chromosome, okay? The centromere standing is absent in the G band pattern. And the C band are associated with heterochromatin along the chromosome and around the centromere. Okay, that's the C bending. And then from this, the actual bending here, the C bending, then you will be able to see the location of the centromere. Okay, and there are two different. You can from the C bending, you can distinguish the, you can identify the mutation. So like the pericentric inversion or paracentric mutation. Pericentric mutation basically involve the inversion that involve the centromere region. All right, that's called paracentric inversion. And then the paracentric inversion, that's the inversion of the sequence that does not involve the centromere. That's called paracentric inversion. Okay, that's C bending. Okay, and then there are many techniques that you can use to detect the genome and chromosome mutation. So one is the karyotyping, right? So you arrest the cell in metaphase, and then you disrupt the nuclei using the hypotonic solution, so that those 23 pair of chromosomes can be assembled in an organized display based on their size. All right, and then you'll be able to see the G bending. And then a different type of chromosomal mutation. All right, so basically involve this alteration of chromosome structure. So like this translocation, right? Translocation, the whole piece being translocated to different region. Deletion, remove one piece of the chromosome, right? Inversion, you inverse a region of the chromosome, like here AB, and then invert to BA. Isochromosome, basically from the one arm from each chromosome and then link together. That's isochromosome insertion. So another piece of the chromosome region inserted into this chromosome. That's insertion, or they can make a ring, so it's a ring chromosome. Or derivative chromosome, that's another piece of chromosome region attached to this normal chromosome. That give you 3 pieces here. That's derivative chromosome. Okay, so these are commonly seen chromosomal mutation, translocation, deletion, inversion, isochromosome, insertion, ring chromosome, derivative chromosome. And this is the genetic abbreviation. Okay, so for example, if you see DEL, that means deletion. DER, derivative. Duplication, insertion, inversion, isochromosome, maternal origin, paternal origin, and ring chromosome is R. Translocation is T, and TL is a telomere. All right. So these are some abbreviations that later you will see there's some example. Okay. Then another technique that people use to identify the chromosome. Now you can locate the location of a specific gene on the chromosome. It's called a in situ hybridization. Okay, in situ hybridization basically you collect a cell, all right, and then you fix the cell in metaphase and put on the slide and lyse the cell, right? You use a formamide to denature the DNA, so it become single-stranded, and then you add a probe, a labeled probe, either the isotope labeled probe or the fluorescent labeled probe. And then this probe is a specific gene that you want to identify. Then you hybridize with the chromosome on the slide. Then you'll be able to see the location of that specific sequence. That's called in situ hybridization. And then if the probe, you use the fluorescence probe, then that become a fluorescence in situ hybridization fish. Okay, and that's called fish. in situ hybridization. So if you use the fluorescence labeled probe, okay, and you denature become single-stranded, and then you mix with the chromosomal DNA on the slide, right? Since you treat this cell with the alkaline, the sodium hydroxide, for example, to increase the pH, so the DNA denature, and then those probe will be able to find the complementary sequence, and then bind it, and then give you a signal, so that you'll be able to see the location of that specific sequence. That's a fluorescence in situ hybridization, is also called FISH. Okay, again, when you prepare these for the metaphase FISH sample, this is metaphase, all right? The metaphase FISH sample, you begin with the culture of the cell. So you have the cell culture for 3 days, and then about 4 minutes before you harvest the cell, you add the cosmic. The cosmic basically will arrest the cell in the metaphase stage, all right? And then you add the hypotonia media, so like a 0.075 molar calcium potential chloride, 0.075. Normally the cell concentration is a 0.9, and if you use hypotonic, so that's a 0.05, 0.075 molar potential chloride, and then you fix the cell with the methanol acetic acid, and then the fixed cell suspension applied to the slide, and then let it dry. Then the target DNA in cell is fixed and denatured on the surface slide. Then you can add a probe to find the complementary sequence, so that you can identify the presence and location of that specific DNA probe. So that's the fluorescence in situ hybridization FISH. And then another FISH we call it interface FISH, right? Interface FISH. Since the metaphase FISH, you need to grow a cell interface FISH. Cell culture is now required in the interface FISH. So this technique normally used for the prenatal samples, tumor, and the hematological analysis. Okay. And to prepare the interface FISH, it will be important that the critical step is in permeabilization the cell for optimum probe target interaction and maintaining the cell morphology. Okay, that's the interface FISH. Okay, so briefly, for the interface FISH, right? Before the DNA denaturation, the cells are dehydrated in greater concentration of ethanol, and then you can use it, right? It's interface FISH. And then to identify the specific region of the DNA in the chromosome, we can use a different type of probe, right? Marina, yeah, what happened, Marina? FISH. Yeah, it's called FISH, exactly. And the 3 different type probes are commonly used, right? So we can have the whole chromosome painting probe, the WCP probe. So the WCP probe basically are designed to mark the entire chromosome of interest. And WCP probe are useful for deciphering cytogenetic aberration that are difficult to resolve on morphological grounds. Okay, so that's the WCP probe. Also, we have this repetitive sequence probe. That's for the chromosome enumeration. And the centromere enumeration probe, CEP, basically is commonly used because hybridized to the alpha satellite or beta satellite repeat sequence within the specific centromere region of each chromosome. We can use that for the chromosomal enumeration. And CEP can identify the aneuploidy of any chromosome. And local specific identifier, LSI probe, that can be used for gene fusion, gene deletion, or duplication. Okay, that's local specific identifier. So this is the example of the WCP, whole chromosome painting. See the whole chromosome painting, the chromosome 6, all right? Also, they have this centromere probe of chromosome 10, right? So this is the centromere of chromosome 10, it's labeled. And also the WCP can be used in the analysis of sperm chromosome segregation in the man, okay? And the man with the heterozygous for the uncommon Robertsonia translocation. And this translocation can result in a reproductive failure because of imbalance in chromosome melted segregation. Okay. And here we have these two the whole chromosome painting probe, right? Including the chromosome 13 WCP, right? And also the WCP 22, right? Then the green one, you see this green one that give you the WCP 13, and then the orange one will be WCP 22. Okay. And here is just to show you the application of fish for the pattern, like for enumeration probe that you can use to quantitate the amplification or deletion. Okay. This here, the gene enumeration, the probe, right? And then you can see this amplification normally should be 2 and 2, and then you have extra 3, that's amplification. Then you lost 1, that's deletion. Okay.
So wait, Professor, so are you amplifying the specific gene that you want, like the red?
No. You label it. You label it, and then if it's you label with the gene enumeration probe. If it's a normal, then this is what you should see. If you see this, that means that's the duplicate of the specific chromosome. If it looks like this, that would be the deletion of a specific chromosome. That's what you see.
So is it like a marker? So this is like the top one is the normal
marker. Yes, the marker, exactly.
So if you have too much of it, that means that the gene is amplified too many times.
Correct.
Okay.
Right. So that can determine and quantify the extra chromosome or the deletion of chromosome. And then for the other one, the different probe, the fusion probe, right? You can identify that specific gene fusion, right? So for example, the normal chromosome look like this. If you see these two attached to each other, so that would be the fusion, right? Yeah. Or originally look like this. Then if you observe like this, that will be a break event. Yes, what's your question, Marina?
When you amplify the DNA,
no,
isn't
we
it?
don't amplify. We don't amplify. This is just the probe, the use.
Okay. That's what we're targeting.
Yes. So basically, it's on the top, That's why you should see the normal chromosome like this. If
Okay.
you see chromosome like this, that red one has been amplified. If you see the red one like this, that'll be a deletion of a red chromosome.
Okay, I see. I was
Okay?
gonna say if we're amplifying, we would want to amplify the genetic
No, we
material
don't
because that's more
amplify.
sensitive.
This is just a chromosomal defect. If you see this, that's a chromosomal defect. This
Okay,
is
I see.
normal.
I was thinking
Okay.
like if we're doing a test, like we want to amplify the DNA because of it would make the diagnostic test more sensitive and then give us, a higher
No, we
chance
don't.
of it being correct.
We don't do any amplification. You just
Okay.
add the probe.
No, I
and
see
then
now what
to
you're
see-
saying. Okay.
Mhm. And then here, that's interface fish, right? You can see these are the centromere enumeration probe. Okay, so it can hybridize to the alpha or beta satellite repeat sequence in the centromere region. Okay, and then so you will be able to see the aneuploidy of any chromosome. So this is just this example here, right? This example that you can see whether you have extra one or you're missing one, right? And this is another interface fish. And this one, we use the local specific identifier probe. So you can identify the aneuploidy of chromosome. And here you can see A, that's 2 green signal, right? And B, you see the 3 green signal, right? And this one is a disomy 2, and this is trisomy 2. So that means the chromosome number 2. For this sample, you have extra chromosome number 2. Okay, extra chromosome number 2. So that's interface fish. And then of course we have this the dual color system. So you have two main local specific identifier fish probe system can be used to detect the gene rearrangement, and the dual color translocation probe and dual color break apart probe that you can use, right? So this is just to explain here, right? We can have this amplification, deletion, fusion, break apart. All right, so from all these analysis, then you will see the difference. And then this is the telomere probe. So it's useful. The telomere probe is useful for detecting chromosome structural abnormalities, like the cryptic translocation, small deletion. All right, so that's a telomere probe. So in this panel A, basically this is they label this 21 long on telomere, okay, 21Q telomere, and also we have the 21Q telomere is a D21 S1446. Okay, I don't see where it is. And then also we have SE1321. SE13, this is SE21, SE13, right? SE13, right? So basically you can use these to look at the deletion of the subtelomere region, okay? And this is the whole chromosome painting 21 and 22. See 21 and 22. So that's basically the fish technology, and those 3 different type of probe that can be used to identify those chromosomal defects. Okay, and then for the advanced fish technology, so basically they are main methodological advance in molecular cytogenetic technology. Okay, so we have this comparative genomic hybridization. It's called CGH, the spectral karyotyping SKY, and multicolor fish, it's MFISH, multicolor bending MCB, and the array comparative genomic hybridization would be the array ACGH. So all these cytogenetic techniques add color to the monotonous world of the conventional bending. Okay, so like this one MFISH, so the MFISH and Sky, these are two multicolor fluorescence technology. Okay, they are the spectral karyotyping method. So you use the fluorescence dye that can bind to the specific regional chromosome, and then you using a series of specific probe, each with a varying amount of a dye. Different pairs of chromosomes show the unique spectral characteristic. Okay. For the MFISH, right? For the MFISH, basically, the MFISH combine the binary ratio labeling and the color changing karyotype. And this method offers the capacity to hybridize the simultaneously 24 or more probe. Okay. And based on the simultaneous hybridization of 24 chromosome-specific pending probe, right? And the label with the different combination of 5 fluorochrome, okay? So in the MFISH, a highly sensitive monochrome camera captured a series of images that have passed through the different filter, okay? That's MFISH. And the SKY Sky, based on the principle of spectral imaging and the Fourier spectroscopy. So you use 5 pure dyes that are spectrally distinct. You use in combination to create a unique chromosome cocktail of the probe. Okay. And both the Sky and the MFISH, they require the use of the human whole chromosome pending probe, WCP probe, so that you can see the whole chromosome probe on this figure. See this, the whole chromosome, all right? Because they use the 5 different chromochrome, so they have different combination. Then they show you this different color, and from this color, then you'll be able to visually identify the mutation immediately. That's MFISH and Sky. Okay, and this multicolor bending MCB. Okay, so multicolor bending MCB is a high-resolution molecular cytogenetic technique for the analysis of metaphase chromosome. And this technique involves the micro dissection of the chromosome loci to obtain a set of probes that produce the multicolor, the Shu-G bending, and therefore the MCB DNA probe contains a mixture of region-specific partial chromosome pending probe, PCP, right, and labeled with 3 to 5 different fluorophore. Okay, so just like this example of MCB, right? And then you can see the immediately, this chromosome number 18 is a ring chromosome. Okay. And then the comparative genomic hybridization, CGH, okay, is an efficient approach to a genome-wide screening for chromosomal copy number change, all right? You can use this to determine the chromosomal copy number change. So basically, you have this metaphase chromosome, and then you can do this metaphase CGH, okay? That can do the genome-wide analysis of cancer specimen with chromosomal aberration, okay? They are either too many or too complex to be fully characterized by the rough cytogenetics. Okay. And for this cytogenome, the comparative genomic hybridization, basically you collect the cell, then immobilize the cell and denature the normal chromosome, and then you use the test, the DNA probe or the reference DNA probe. Then you hybridize with the chromosome that you collect from the clinical specimen. Then you'll be able, from the signal, you will be able to determine whether that specific DNA is amplified or deleted. Okay, that's a CGH, right? So basically, you immobilize the DNA, the chromosome, and then also denature it, and then you have the labeled DNA probe hybridized with those chromosomes, and then you will compare the signal. So if you have the amplification of particular DNA region, then you will see these strong signals, like green, the test the color. If you see red, that's deletion. Okay, and then you can go through this whole chromosome to compare the test to the reference ratio. If it's a 1, then that's normal, no change. Then if it increased to 1, more than 1, that's amplification, all right? And if it's less than 1, that's deletion. You just compare the signal when they bind to the particular region. That's called CGH. And just like this, so you have this CGH, and then compare the signal and give you this, and you will see here is below 1, so you know the deletion in this region. Okay. So basically, you have the tested DNA based collect from the patient, and then you have normal reference DNA, and both DNA are labeled different label with different fluorochrome. Okay, and then you hybridize to this normal DNA chromosome, normal chromosome spreads. All right, and then you compare their signal. So if the normal reference DNA, the amount of the normal reference DNA is similar to the amount of a test DNA, then that should give you the ratio 1. Then you don't see any color change. All right. If the reference, okay, is less than the test, so the test has been amplified in that particular region, then you should see the red signal. If you see more reference and less the test, so that means that region has been deleted. Okay. That's how you compare the ratio. That's called a CGH. And then another technique is called array CGH. Right? The CGH already increased the resolution. However, it's still not very accurate. And now the scientists developed this array CGH. Array CGH, that means the reference DNA, basically they collect the chromosomal DNA, all right? Not just like what you see here, you use the chromosome as a reference, but now you purify the genomic DNA, all right? Those genomic DNA you spray on the slide according to the location, okay? And this is time, and then you hybridize with your test and reference probe, and so that you will be able to more accurately identify which location, which region of the sequence has been, for example, in here, deleted. All right, the ratio is less than 1. Okay, so that's the deletion in this region, right? And all the rest region are around 1-to-1 ratio. Okay, so that's array CGH. So in array CGH, that involve the substitution of hybridization target, the metaphase chromosome with genomic DNA sequence. That genomic DNA sequence adhered on the glass slide, right? And so you can increase the resolution for detecting copy number changes in the human genome. And then you can see the gain or loss of the specific DNA. So this is the ACGH. And this, the advantage to use the FISH in clinical diagnosis, basically significant advantage of FISH is can be applied in non-dividing cell, and then facilitate the direct investigation of chromosome in cytological preparation and tissue section. Okay, CGH. And the chromosome disorder basically is caused by the abnormalities in the number or the structure of chromosome, and the cytogenetic karyotyping is a standard practice to identify all chromosomal disorder. Okay. One of the examples would be the abnormality in which the chromosome number is an exact multiple of the haploid number. It is larger than the diploid number. We call it polyploid. And polyploid arises from the fertilization of an egg by two sperm, or the failure in one of the division of either the egg or the sperm, so that the diploid gamete is produced. The aneuploidy basically occur when the chromosome number is not an exact multiple of the haploid number, and result from the failure of a paired chromosome to separate at the anaphase. Okay. And then for this one, the chromosome number of abnormalities, aneuploidy. So you see the 48, XX, XX. So you have 48 chromosome. Normally you should have 46. Now you have 48. And those two extra chromosomes are from sex chromosome, XX. Okay. So that you can see you have 4 X chromosomes. So that's called 48 XX, XX. And then for this one, the trisomy 21, so that means 21 chromosome 21 has 3 chromosomes. So you look at 21, you have 3 here. So it's a 47 XX, XX, that means female, plus 21, that means this one extra chromosome is on chromosome number 21. Okay. And the 45
do these chromosomes cause any like diseases?
Yeah. Yeah, like a trisomy 21, I believe it's a Down syndrome. Okay. And then this is the 45 XY, and then translocation 14Q to 21Q. Okay. And 41, right? And then, no, 45, and then XY male, translocation T, 14Q, right? From the 14 here, right? Translocation to this 21Q. Okay, that's translocation. And the reciprocal translocation, so for example, you can have 46 XY translocation, the reciprocal translocation between 2 and 11, and the exchange between P21 region and Q23 region. That's called reciprocal translocation. And then you see this extra copy of the chromosome number 5. And this is the example. Chromosome number 4, see green R, green number 4, 46 XX female. And deletion, okay, there's a syndrome deletion here. There's a 4p deletion here, right? The deletion number 4p16-1, the region, the subregion 16.1 in the PR. Okay, deletion number 4. Inversion. So, for example, you have inverted this region 5Q 15.1 to 13.3 have been inverted. So that's called inversion. And that causes this Hermansky-Pudlak syndrome too. Okay, the derivative chromosome. So you can see here, XY, here, 16, it's mouse. It's my mouse here. 46 chromosome XY male, derivative 11. All right, derivative 11, translocation between 6 and 11. Okay, P22.1 and P15.2. Okay. All right, so that's it for the chapter 13. Any question?